This video is going to pick up from the end of glycolysis and talk about two processes that take place in the matrix of the mitochondria, and these are the link reaction and Krebs cycle. So we're going to start off with the link reaction. Link reaction uses pyruvate to form acetyl groups. Pyruvate, which is the product of glycolysis, contains a lot of chemical potential energy, and that can be released further in the chemical reactions of the link reaction and Krebs cycle. Pyruvate is actively transported into mitochondria and travels to the matrices, the matrix of the mitochondria. So if we imagine that we have a mitochondrion, for example here, and we've got the outer membrane, and this here is the inner membrane. The inner membrane has lots of foldings that form what we call cristae. In the cytoplasm, the process of glycolysis has taken place. So we've got glucose going to form pyruvate through the process of glycolysis, and then that pyruvate is actively transported into the matrix of the mitochondria. And it's in the matrix, as I've said, where the link reaction and then Krebs cycle takes place. Pyruvate undergoes a reaction which actually has two parts to it. Pyruvate is converted into an acetyl group, and that acetyl group then goes on to the next stage. During this process, two reactions take place. The first is the removal of hydrogen, and that is given to NAD to form reduced NAD. So once again, NAD is acting as a hydrogen carrier. It is a coenzyme. Because that process involves the loss of hydrogen from pyruvate, it is described as an oxidation. Now, the term oxidation can be misleading, so we could also call it a dehydrogenation. The other main chemical event that takes place is that pyruvate loses a molecule of carbon dioxide, and we call that a decarboxylation. So D means remove, carboxy means carbon and oxygen. So we're removing carbon and oxygen from pyruvate. And this is a good time to come back to pyruvate, and remember that pyruvate is a 3-carbon compound. So if we are removing hydrogen here, and then we're removing carbon dioxide here, we are removing a carbon atom. So acetyl is actually a 2-carbon compound. Now, acetyl groups do not have all their bonds filled. There is actually an extra bond that doesn't have anything attached to it. So acetyl groups on their own can be quite dangerous. They need to be joined onto something, and if they are not joined onto something, they'll get lost, or they might attach to another molecule and cause some sort of damage there. So what we have coming in to the link reaction is another coenzyme, and its job is basically hold on to acetyl groups and then release them. And that coenzyme has got an easy name to remember. It's called coenzyme A. So what happens is these two processes take place, which forms the acetyl group. Coenzyme A comes in and joins on to the acetyl group. So the product of the link reaction is actually acetyl coenzyme A, and we say that is one substance, but in fact it is where the next process is going to take place, because acetyl coenzyme A will go off and the coenzyme A will lose the acetyl groups, and we'll talk about that in the next part. We just go back finally to this process. So we have got an oxidation and we've got a decarboxylation going on at the same time. You'll see very shortly this is quite a common series of reactions. And so we call that reaction an oxidative decarboxylation. But remember, even though there's an oxidation taking place, that oxidation is due to the loss of hydrogen from pyruvate. Now, that is the link reaction, and it takes place in the matrix of the mitochondria. It is a stage where fatty acids in the diet can also enter respiration. So we've been concentrating on respiration starting with glucose, starting with hexose, a carbohydrate. But cells can also use other molecules as a source of energy and respire them. So the link reaction is somewhere where fatty acids in the diet can enter respiration. And if you remember, fatty acids have got long hydrocarbon tails, and they can be sliced up and converted into acetyl groups. So if we've got a fatty acid over here, so there's our fatty acid with hydrocarbon tail. What basically happens is that hydrocarbon tail is cut up into smaller parts, and they are converted into acetyl groups, and then they are picked up by coenzyme A, and that means that fats, lipids can be used as a source of energy. They miss out glycolysis, and the link reaction, they go in straight here and then carry on down to the next part. Okay. Amino acids can also be broken down and enter at this point as well. The detail of this you don't need to know. So finally, let's go back to this process and remember that in glycolysis, one molecule of glucose actually produced 2 molecules of pyruvate, which means for every glucose or hexose molecule that enters glycolysis, the link reaction is going to occur twice, because you get 2 pyruvate molecules produced from one glucose molecule. So the next part is finding out what happens to acetyl groups, acetyl coenzyme A, when they go to the Krebs cycle. after the link reaction, the acetyl coenzyme A that is produced then moves on to the Krebs cycle, which also takes place in the matrix of the mitochondria. So we had from the other reaction, acetyl coenzyme A from the link reaction. This splits into coenzyme A and acetyl groups. Remember, these are two carbon molecules. The coenzyme A will then go back to the link reaction to be used again and pick up a new acetyl group. So coenzyme A is constantly being recycled. Now this takes place at the same time as other reactions that are taking place in the Krebs cycle. So remember, in the matrix of mitochondria, these substances are moving around and flowing between each other, but we will draw them as a circle to see what happens and the importance of it being recycled. So acetyl groups move on to the Krebs cycle, and acetyl groups join to a 4-carbon compound which is in the matrix of the mitochondria. And this 4-carbon compound, you do not need to know the name of. And those acetyl groups join onto that 4-carbon compound. So one acetyl group will join onto a 4-carbon compound. So of course what we get formed is a 6-carbon compound. And once again, you do not need to know the name of that compound. What happens next is a series of chemical reactions that eventually lead back to that 4-carbon compound. But in the process, the hydrogen atoms that are in the acetyl groups are slowly going to be removed and given to coenzymes, which later on will be used to release energy to make ATP. But that'll be the next stage. The first reaction that takes place is something which hopefully you will be familiar with, and that is the loss of hydrogen to NAD to form reduced NAD, and that is a source of chemical potential energy which we'll use later on, and the loss of carbon dioxide. This is exactly the same as the link reaction. It is an oxidative decarboxylation. Exactly the same process as in the link reaction. And of course what we get formed therefore is a 5-carbon compound because we've lost a carbon from there. And then the same reaction takes place, exactly the same. So NAD comes in and we get reduced NAD formed with the loss of hydrogen. So another point where hydrogen is being removed from the acetyl groups, source of chemical potential energy, and the loss of carbon dioxide. So what that means is oxidative decarboxylation occurs twice. And the result, of course, is that we will have a 4-carbon compound produced there. This 4-carbon compound then goes through a series of chemical reactions, but we end up with a 4-carbon compound there, which means that we don't lose any more carbons, we don't gain any more carbons, and all the reactions that are taking place involve the other atoms in these molecules. So they'll be mainly oxygen and hydrogen. So the first reaction that occurs next is a rearrangement of that 4-carbon molecule. And the rearrangement of that 4-carbon molecule releases energy. And that little bit of energy that is released there is just enough to join an ADP molecule to an inorganic phosphate molecule to form ATP. And this is an example, again, of substrate level phosphorylation. Substrate level just means at the point of these molecules reacting and turning from one into another, as opposed to the process that we'll look at in the next lesson. So I'll come back to this in the next lesson. We end up with a 4-carbon compound again, and then that is converted into another 4-carbon compound. And to do that, we need to lose hydrogen again. However, for some reason, NAD cannot take the hydrogen away from this molecule. A different coenzyme is needed, and this other coenzyme is called FAD. It's exactly the same process. FAD comes in, picks up hydrogen from this 4-carbon compound, and we get reduced FAD formed. It does make a difference, you'll see that next time, but generally the process is exactly the same. So we get another 4-carbon compound formed. This 4-carbon compound is then converted into the final 4-carbon compound. And to do this, this 4-carbon compound has got to lose hydrogen again. And then at this point, NAD can come back in, so we can form reduced NAD. And that, of course, is important for later on. And that is the Krebs cycle. Now let's just have a quick overview of what is happening. Acetyl groups come in from the link reaction. These acetyl groups are made of 2 carbon atoms, some hydrogen and some oxygen, but you don't need to know the detail of that. What happens then is that those acetyl groups are attached to a 4-carbon compound, and then that slowly goes through a series of reactions where the 2 carbons that came in, are lost, and the hydrogen that's in the acetyl groups are slowly removed. So some hydrogens removed here, some hydrogens removed here, some is removed here, and some is removed here. And the process is taking away small groups containing chemical potential energy, which then can be used by the next stage to release energy to make ATP. Now, you don't need to know the names of any of the processes involved, of the molecules involved in this process. You do need to know the number of carbon atoms that are present, and that this is oxidative decarboxylation, this is substrate level phosphorylation, and these other processes involve the removal of of hydrogen. So these processes the removal of hydrogen, and I'll highlight those as well. These processes we say are oxidations. But we can also describe them as dehydrogenation, because oxidation can be defined as the gain of oxygen or the loss of electrons or the loss of hydrogen. But here it's the loss of hydrogen. Because in Krebs cycle 2 pyruvate molecules were made, the link reaction happens twice. So you get 2 acetyl coenzyme A molecules produced, which feeds 2 acetyl groups in it, one at a time. So for every glucose, every hexose molecule that enters glycolysis, the Krebs cycle turns twice. So the main parts that the exam board want you to know about for Krebs cycle is that an acetyl group joins with a 4-carbon molecule to form a 6-carbon molecule. That undergoes a series of oxidation reduction reactions in which carbon dioxide is lost, decarboxylation, hydrogen is removed to form reduced NAD and reduced FAD. So we get reduced NAD here, and reduced FAD there. Some energy is lost to form ATP from ADP and inorganic phosphate. And remember, you can't just say to form ATP, you must say from what. And this is the really, really important bit as well. For every biochemical cycle, the starting molecule must be regenerated. So the next part of respiration is what is going to happen to all of these reduced coenzymes, the reduced NAD and the reduced FAD. And these are all going to go to the next stage, and the next stage, which we'll talk about next time, is oxidative phosphorylation.